Thank you for including me in this meeting. I've always been inspired by CARTA as an organization and the people and ideas you bring together around one of the fundamental questions, how we became human. We often discuss the benefits of large brains, but today I want to highlight some of the costs and share with you how we're using organoid models to explore trade-offs in human brain evolution. Dramatic changes in brain structure and function evolved along the lineage leading to humans. Our brains tripled in size in just the last few million years. In my training, I focused on genetic, molecular, and cellular mechanisms that could drive cortical expansion in the human lineage. However, one of the special properties of mammalian brain evolution is that not all brain regions change equally as the brain expands. The neocortex, underlying white matter, striatum, and cerebellum have increased disproportionately compared with the olfactory bulb and brainstem regions, including the ventral midbrain. In my own group, we've become interested in how this process of unequal scaling, combined with new functional requirements, may drive cell type specific adaptations to our large brain cellular ecology. We think some of these trade-offs between forebrain expansion and connectivity may be most strongly manifest in the ventral midbrain. Here, a small cadre of a few 100,000 hardworking dopamine-producing neurons project to vast target regions in striatum and cortex. Since our divergence from old world monkeys, these target regions have increased in volume 6 to 17-fold, but the number of source neurons has only doubled. In addition to these increased demands from unequal scaling, the innervation density of these neurons has actually increased in humans, even compared with chimpanzee in the medial caudate and nucleus accumbens. The dopamine-directed striatum hypothesis predicts that this increased connectivity was actually crucial for the evolution of cooperative behaviors in our lineage. The neurons themselves also have extraordinary specializations. Their axonal arbors span nearly a meter, even in rodents, creating enormous energetic demands that increase oxidative stress and vulnerabilities, particularly in nigrostriatal neurons that undergo energetically expensive pacemaker activity. And the production of dopamine itself further releases reactive oxygen species, making these neurons particularly vulnerable as connectivity demands increase with brain expansion. As such, it may not be surprising that these neurons are implicated in a number of disorders enriched in humans compared with other primates, most notably including Parkinson's disease caused by the loss of the nigral population. So although these neurons represent a phylogenetically ancient vertebrate population, we reasoned that new demands in the human lineage may have spurred recent adaptations, and we sought to investigate how dopaminergic neurons have evolved in response to brain expansion and new functional requirements. And specifically, we were interested in identifying molecular factors that could inform the developmental origin of connectivity differences, as well as exploring whether compensatory adaptations evolved in response to the increased demands these neurons face in the large brain cellular ecology. And how do we tackle this question when experimental access to human innate brains is limited for ethical reasons? We were fortunate to have a talented postdoc, Sarah Nolbrandt, join the lab. Sarah extended pluripotent stem cell models of dopaminergic neuron differentiation and maturation from human to across the primate phylogeny in 2D, and further adapted these to 3D organoid models. This allowed us to compare dopaminergic neuron development and gene expression between species. To isolate species differences and mitigate batch effects, Sarah scaled the experiment across many individuals in a pooled culture format within each species, often described as a village in a dish. But Sarah also went further and generated interspecies organoids with cells from every individual and species represented in a common garden environment in order to isolate cell-intrinsic molecular differences between species and further reduce batch effects. Here's an example of what one of Sarah's experiments looked like. Here we've pooled together IPS lines from 17 individuals across 4 species, exposed these to dual SMAD inhibition to generate neurectoderm, activated Wnt signaling to form midbrain, and applied sonic hedgehog to ventralize these. By day 16, these cells form ventral midbrain patterned progenitors that can be cryopreserved and reproducibly thawed to form midbrain neurospheres. These neurospheres produce dopaminergic neurons with axons expressing tyrosine hydroxylase in green, the rate-limiting enzyme for dopamine production. These axons wrap around the periphery and can be matured in long-term culture over 6 months. In addition, when we fuse these interspecies ventral midbrain organoids to target regions, we can see that these Th-positive axons can actually project to the target regions. As the organoids mature, we see spontaneous activity and we see the release of dopamine, showing that we can realize functional maturation in the model system. So next, we wanted to measure gene regulatory divergence in these common garden environments of primate dopaminergic neurons. And to do that, we have cells from each species and individual mixed together, and we needed to find a way to assign these cells back to their individual and species. Nathan Schaefer in the lab developed a computational toolkit that uses naturally occurring sequence variants in the transcript to genotype the cells and assign them to individual, while also removing ambient RNA. And we call this package Cell Bouncer for checking cell IDs and keeping out the riffraff. Using Cell Bouncer on a dataset of gene expression from these organoids, we could identify diverse cell types grouped in this projection based on transcriptional similarity, with each dot representing a single cell. Notably, cells from each species were maintained across the long-term cell culture, and we were able to maintain representation. About a third of the cells, highlighted in blue in the chart below from each species, belong to the dopaminergic lineage, denoted by the expression of LMX1A. In addition, we saw conservation of many marker genes, with a few highlighted here in dopaminergic neurons from each species and as referenced to a primary cell atlas of human brain development. Before going on to compare gene expression between the species, we wanted to gauge whether the cells mature at the same or different rates. And to do that, we could reconstruct the sequence of transcriptional changes during dopamine neuron maturation using an approach called pseudo-time trajectory inference. And applying this approach, we could see the expected genes come up, but we could also score cells from each species. And when we did this, what really stood out is the cells from rhesus macaque mature much more quickly. At day 40, some of the cells have already reached the day 80 stage in the other ape species, whereas we observed more comparable maturation rates in human and chimpanzee. By an orthogonal measure, the switch from neurogenesis to gliogenesis, we could also see a similar pattern where rhesus macaque switched to gliogenesis much earlier, supporting these the molecular staging of maturation. We could then apply these homologous cell types and quantitatively defined maturation stages to ask how have homologous cell types diverged in gene expression using a linear mixed model to account for this variation. And when we do that, we can see that gene expression is largely correlated between species in dopaminergic neurons and other cell types, but we can also identify gene expression differences that evolved specifically in the human lineage using the other species, rhesus and orangutan, as outgroups. And when we do that, we can further validate some of these examples, like this inward rectifying potassium channel, KCNJ16, that shows very specific expression in human dopaminergic lineage cells, but not in other species. In addition to looking at gene expression, we were also able to measure chromatin accessibility in the same cells. And we can see a concordant increase in chromatin accessibility at the promoter of this gene in human, further supporting the gene expression difference. Using this approach, we identified many candidate genes, including those that could influence connectivity, such as neuritin-1, a neurotrophic factor that promotes axon outgrowth, with a human-specific increase both in chromatin accessibility and gene expression, specifically in the human lineage. Across divergent loci, we observed an enrichment for human-specific structural variants overlapping differentially accessible regions. For example, at this GABA receptor locus, we observed a human-specific insertion overlapping a non-coding cis-regulatory element and appearing to disrupt and reduce the chromatin accessibility. And this was concordant with reduced expression of the nearby GABA receptor gene. Looking broadly among the upregulated genes revealed that most strongly divergent categories in immature neurons related to connectivity. including a range of neurofilament genes and genes linked to axonal transport that were upregulated specifically in the human lineage. When we looked at more mature dopaminergic neurons in our culture system, remarkably, we observed that the most strongly upregulated categories actually related to oxidative stress buffering, consistent with our hypothesis that compensatory mechanisms may have evolved in this lineage. And this included a gene catalase that was recently shown to be upregulated in adult human cortical neurons compared with chimpanzee and rhesus macaque, supporting the in vivo relevance of some of these in vitro models. Although we can't age these organoids for decades, we can expose them to oxidative stress acutely and then directly examine whether human-specific regulatory networks, such as those we just identified, influence the stress response. And here, we can really leverage the phylogeny and a dish approach by evaluating these cross-species differences in a controlled common garden environment where cells from both human and chimpanzee are represented together in the same state. By adding rotenone, which blocks the electron transport chain, we could recapitulate the selective vulnerability of these neurons, the specific loss of TH in these pictures, and then we could measure gene expression across individuals using cell bouncer. And when we do that, we see that the major axes of variation relate to the stress response, principal component 1, and to species differences, principal component 2, with parallel response trajectories in human and chimpanzee. And these response trajectories include conserved bioenergetic stress genes. For example, heat shock proteins, early response genes, and mitochondrial genes are upregulated in both human and chimpanzee, while neuronal marker genes are downregulated. In addition to these conserved responses, we could also see that the oxidative stress stimulation unmasked species-specific differences in the response to oxidative stress, including BDNF, which was transiently upregulated in humans, but not chimpanzee, and the mitochondrial calcium uniporter MCU, which was transiently downregulated. We were excited about this because both of these genes, the activation of BDNF and the repression of MCU, have been shown to promote dopaminergic neuron survival in Parkinson's disease models, consistent with evolved neuroprotective effects in the human lineage. Given the baseline increase we observed in oxidative stress buffering genes and these human-specific stress-dependent responses, we next asked whether the trajectory of oxidative stress response differed between species. And when we did this, we found that all 7 chimpanzee individuals had stronger responses along this molecular trajectory than all 8 human individuals, suggesting that even in this simplified in vitro model with young dopaminergic neurons, the stress response may be blunted in human cells by compensatory molecular changes. To conclude, we generated interspecies ventral midbrain organoids using a phylogeny and a dish approach that produce dopamine and resemble primary brain atlas dopaminergic neurons. By comparing gene expression and chromatin accessibility in a controlled environment, we were able to identify human-specific changes and candidate genetic mechanisms driving these alterations. The top-ranked categories in immature and maturing dopaminergic neurons related to changes in connectivity by changes in neurofilament genes and axonal transport genes, as well as changes in antioxidant activity and oxidative stress buffering, including peroxyredoxins and catalase. Further, by applying acutely in this experimental model oxidative stress stimuli, we could unmask human-specific neuroprotective responses and see that the human cells had a blunted overall response compared with chimpanzee across many individuals. So coming back to our early question of whether dopaminergic neurons have evolved molecular changes in response to trade-offs in human brain evolution, we see evidence that there is, in fact, an increase in buffering of oxidative stress response that may partially compensate for the dramatic changes in connectivity that have evolved in our larger brains and with new functional requirements. So even though this is a simplified system, it shows us how we can investigate cell type evolution and the response to trade-offs in otherwise inaccessible human and ape cell types using organoid models. In the future, we'd be interested to extend this approach beyond long-range connectivity that I focused on here to many other potential costs of large brains. For example, large brains take longer to develop, and humans particularly have a protracted period of development, including a human-specific stage of early childhood, where there's dramatic changes in neuronal morphology. In addition, large brains create new challenges for vesicle trafficking, bioenergetics, and the reorganization of neurons, reducing average connectivity between them, creates modularity and computational constraints that could be investigated. So in closing, evolution involves trade-offs, and we can combine organoid models and single-cell genomics to learn from how cells and systems have responded to these costs from a molecular perspective. And I want to thank a number of people in my group who really made this work possible. Sarah Nolbrandt established this system. Janelle Wallace led the gene expression divergence analysis, and Jing Wen led regulatory network analysis I didn't have time to talk about, while Nathan Schaefer and Brian Pavlovich established the cell bouncer package, and Brian reprogrammed many of the ape lines that we used in this study. So thank you for your attention, and I'm so grateful to be part of this symposium.